Quick Detection Guide for Citrus Huanglongbing Candidatus Liberibacter asiaticus. Sample processing. You'll need field isothermal fluorescence, rapid detection kit for Citrus Huanglongbing Box A, adjustable pipette, 50 microliter range with 200 microliter tips, or 50 microliter fixed volume pipettes, and marker pen and disposable. Open Box A. It contains the instruction manual, grinding bags, sample processing solution, nucleic acid release reagent. Put on disposable gloves. Select leaves to test and remove the leaf veins. Important: change gloves between different samples. For each sample, take 2 to 3 leaf veins. Break into 3 to 4 centimeters piece if too long. Place the veins into a grinding bag. If processing multiple samples, label each bag clearly to avoid mix-ups. Change gloves before handling the next sample to prevent cross-contamination. Take 1 milliliter sample processing solution. Squeeze the dropper bulb first, then immerse the tip below the liquid surface. Release slowly until the solution reaches the 1 milliliter mark on the dropper. If the first draw is less than 1 milliliter, for example, 0.75 milliliter, add it to the grinding bag first. Then draw the remaining amount, for example, 0.25 milliliter, to complete 1 milliliter. Seal the grinding bag. Place the bag with the mesh side up. Use a pen or other firm object to grind until the vascular bundles are exposed and the solution turns green. Now let's move on to nucleic acid release. Take the corresponding number of nucleic acid release reagent tubes and label each tube. Take the pipette and tip. Mix the ground solution by gently squeezing the bag toward the sealed end. Tilt the bag and carefully draw 50 microliter of the processed solution. Add the 50 microliter solution to the nucleic acid release reagent tube. Cap the tube, mix by shaking, and let it stand at room temperature for 5 minutes. We can also use a 50 microliter fixed volume pipette. Carefully squeeze the ground solution toward the sealed end. Squeeze the bulb before inserting the tip into the liquid. Release to fill the tube. A small amount may enter the overflow chamber. Dispense the liquid from the delivery tube into the release reagent tube to finalize 50L sample addition. Nucleic acid amplification. You'll need field isothermal fluorescence rapid detection kit for Citrus Huanglongbing, Box B, digital isothermal detector, adjustable pipette 50 microliter range with 200 L tips. Open Box B. It contains the Citrus HLB isothermal fluorescence detection reagent, negative control and positive control template 099. Instruction manual. Prepare 1 HLB detection reagent tube per sample, plus extra for positive and negative controls. Using a pipette, add 50 microliter of the sample nucleic acid into each reagent tube. Change tips between samples to avoid contamination. Add samples, positive and negative control, into the reagent. You can use a reagent tube rack for a large number of samples. Cap each tube immediately after adding the sample. Tubes are numbered. Note the tube number and match it to the sample. Invert each tube 8 to 10 times to mix thoroughly. Then briefly centrifuge or tap to collect liquid at the bottom. Turn on the digital isothermal detector. Place the reaction tubes into the instrument and note their positions. Close the lid and press the start button. The blue indicator will blink and a 20-minute countdown will appear on the screen. After 4 minutes, a beep will sound. Open the lid and take out the tubes. Invert each tube again 8 to 10 times, mix well, and tap to collect liquid at the bottom. Return the tubes to their original positions in the detector without pressing any button. Close the lid to continue amplification for the remaining 16 minutes. When the amplification finishes, the instrument will beep. Refer to the instrument version and product manual for specific result interpretation guidelines.